All right, so here we are on the 7th, starting a new unit. I just handed out a revised schedule. It moves around some topics, but not some due dates. And let's see, the other thing you're all wondering about is the exam. I will hand that back at the end of class today. No scores are recorded yet, and I need them back from you in order to record the scores. I won't have scores unless you hand it back to me. I don't give, in this class, I don't give the exams back because I tend to reuse similar things from one year to the next. But I hope you all found it pretty similar to the quizzes, and a few homeworks kind of extracted and pulled in here and there. Hopefully wasn't too unfamiliar. And the performance on this one, I don't know if it was record-breaking good, but it was really good. Okay, very, very few concerns on overall scores. Most of the scores were very, very strong. I have no suggestions except just keep doing what you're doing and call it good. All right, so yeah, I'll quit several minutes earlier. And the only thing I'd ask on the exam itself is check your totals. I won't say that I make mistakes a lot, but it has happened enough to make me cautious and ask you to be the second set of eyes, adding up all those numbers. All right, we're headed off into the second unit of the course, and there are, this time, only 2 quizzes instead of 3, or 5, depending on how you count it, I guess. Only 2 quizzes, and hard copies are here, for those of you who like that. They're open on D2L if you like the electronic version. And we're starting out with carbohydrates. So this is, it's a new functional group for us to be worrying with, so let's just do it. The definition of a carbohydrate, I don't know if you ever thought about where this word came from. Carbos is what they tend to be called these days. This is the carbo part, and this is the hydrate. They literally have a formula that makes them look like they are hydrates of water. That's a lot of oxygen. I mean, compared to an alkane or something like that. That's saying that for every carbon atom, there is 1 oxygen atom. That's loaded, okay, which puts them in a different category from fats and other things like that. All right, so they have several different functions, and I'll just put something down briefly about each one of them. The primary fuel would be glucose, and there are some other similar ones that we'll be looking at. A function for, when they function as energy storage, we're looking at, for animals, glycogen. For plants, we're looking at starch. They can also function as something that's not connected to energy at all, and that would be for cell walls, so like cellulose. Humans can't digest that. You'd have to be you can't digest it unless you're a termite. None of you look like termites. And then signaling between cells, this is actually sophisticated, very specialized. This is when carbohydrates are actually conjugated or attached to proteins. All right, so we have several different categories and some terms that go along with each one of them. So you'll need to know these. When we use these terms, we kind of are talking collectively about maybe a bunch of different carbohydrates at once. So monosaccharides mean exactly what they say. There's only 1 unit that you can look at in the whole molecule and go, aha, that is a carbohydrate. That's the simplest version of them. The scale does move up, and there's a set of terms, not a whole lot of them, that kind of collect several molecules at once. This is made up of 2 of your monosaccharides that are joined together. And there's a whole host of chemistry that goes into just the disaccharides. Probably the most famous one for us would be sucrose, because we consume a lot of it. Not all that good for us, perhaps. I sure wish it were better for us. I could eat even more of it and get away with it. Oligosaccharides, maybe 3 to 10. monosaccharides links. And this would here, at the last end of the list, you have, let's say, more than 10. There's this term sugars that gets used. That's probably the least precise, but that's still worth thinking about. They tend to be water soluble and sweet tasting, so things like sucrose for sure you would refer to as a sugar. Fructose, first one is a disaccharide. Fructose is a monosaccharide. And just some general use to it like that. I'll probably throw that around a little bit imprecisely, like the term itself is. But since you've got university background now, you know that carbos and sugars can technically be the same. Depends on how precise you want to be. Okay, so we'll get more precise here by acting like we are somewhat versed in organic chemistry, like we are, like you all did pretty well with your structures. And here's where we start looking as if we're going to put that organic background to use somehow. Functional groups are pretty simpler. Polyhydroxy, so OH, and what they sound like, aldehydes, and ketones. Okay, so there's your set. Now, that's combining several different chapters worth of material from an organic course into one molecule, so they're kind of fun to deal with. I'll just give you a few examples here. About the simplest example that you can have that would count as a carbohydrate would be this. So if you want to start with an aldehyde as a functional group, it would need to look something like this. So that would be what? 3 carbons. 3 carbons, you would call it a monosaccharide. And you would also hopefully recognize that the functional group in this one is an aldehyde. So I guess you could have, you could make that one into a ketone. I'm going to move up into more realistic territory. Let's go to a 6-carbon compound. So I'll say that that's your terminal OH. So 1, 2, 3, 4, 5, 6, and make that the aldehyde. Okay, if you want to throw in numbers here, you're welcome to. I'll put them in. You don't have to put them in. So there's 1 and 6. You don't want to make those too big. But now the definition requires that you have to have the term polyhydroxy. Aldehydes already got 1 oxygen guaranteed. And if you're going to have 1 oxygen for every carbon in the molecule to make it fit the carbohydrate formula, the simplest, most natural way to do that is to go, well, I've got 6 carbons to fill in. I need to make sure that I've got 6 oxygens. So 1, 2, 3, 4, 5, 6. So there you've got it. 6 carbons, still a monosaccharide. And this one is an aldehyde as the functional group. So the functional group you're looking at here, of course, is the one centered on that carbon. Now, you don't have to make much of a change to that structure right there. In fact, you could leave the carbon framework the same. And make sure that we've got 6 carbons. So 6, 5, 4, 3, 2, 1. All the change you'd have to make would be to move the carbonyl group to that carbon right there. So you'd be numbering here, what, 1, 2, 3, 4, 5, and 6. And again, you want every carbon to have an oxygen. So 1 and 2 already do. 6 does. There'd be 4, 5, and 6. Now that's just moving the carbonyl group from the terminal carbon to one of the interior ones, there'd be several different options. If the ketone went to 3, 4, 5, or 6, those would all be constitutional isomers of the first one, meaning all you have to do is look at constitutional isomers, and you've got this world of chemistry, not to mention all the stereoisomers that result if you're counting up the different directions. The hydroxyl is a good point. The stereochemistry just blows up very quickly here, but just for comparison, it might help to say, well, what's different and what's not different between these. The number of carbons sure isn't different. Both of them are also monosaccharides. You can't say that they're different in that regard, but the difference is the second one has a ketone in it, and this is where it's heads up, because you're looking at this one and going, eventually we'll call this one an aldose and this one a ketose. The difference really is where the carbonyl group is placed. If it's on an interior carbon, you're looking at an aldehyde or ketone. Okay, and that's the difference between glucose and fructose. Fructose has its carbonyl group on one of the interior carbons. So I kind of hinted at this already. Some terminology that gets thrown around for these, we'll start with this point right here. An aldose is something that has an aldehyde. The ketose is something that has a ketone in it. Both of these are saccharides, monosaccharides, as we're limiting thing right now. Okay, that much doesn't change. Number of carbons, looks like the list so far we're up to 3 and a 6. So you've seen these already. 3 carbons versus 6 carbons, and then in the middle, pretty intuitive, I hope. Tetrose is something that has 4 carbons, whereas a pentose is something that has 5. And those are terms that get just used. I'll try to pull it all together at some point here, but any of those can be used to describe a sugar, sometimes more than one of them. So I've been on this first page here staying away from stereochemistry, but you can't stay away very long because that's the heart of the whole thing. And so this whole stereochemistry, I was about to say mess, it's kind of a mess, but it's actually good news. It's a source of variety in the compounds. The whole world opens up with the smallest aldehyde you could have and still have a stereocenter. So let's pick that out. It needs to be that carbon right there, right? The other 2 carbons in this 3-carbon molecule, they're sure not going to count as stereocenters. So you look at this first one, you go, well, it doesn't even have 4 different partners. That's not a stereocenter. That's out. The next one, it has all this part of the molecule, that whole end, it's got this whole end, it's got an OH and a hydrogen, 4 very different things. That's a stereocenter. Then you come down here finally to this last carbon in the list, and you're looking at all that chunk of the molecule. That's one, and the OH is the other, but it's got 2 hydrogens on it. So that's not a stereocenter. That's the only stereocenter. So that's the one marked. And this one right here, as it is, keep in mind, this is actually drawn without a stereo view. Okay, all of those bonds are drawn in the plane, meaning that since you've been through organic, you will know that the stereocenter can have 2 different possible conformations. The OH could either be coming out at you or be going back away from you, and those are different molecules when they're viewed by stereocenters. So there's your stereocenter, and remember the reason for it. That stereocenter has 4 different partners. It's the definition of a stereocenter. It's a stereocenter that's also a carbon here. All right, so let's break them down. This one now has a different level of structure. This is an additional layer of understanding of it. When you get to this, you've got something that's now drawn with stereochemistry. Okay, the only difference between the two of them is where the OH and the hydrogen point. Okay, now just to, I'm not going to write this, let's get you on kind of familiar territory. If we're talking about this business called absolute configuration, somebody asks you, points at a molecule, tell me the absolute configuration of that molecule, your answer can be only one of two things. And what are those two things? They're both letters.
S or R.
Yes, S or R. Okay, that's the only possibility. So R or S would be what we'd assign to them, and I don't want to put that on the structure itself because we need them for other things. But let's just do a little review and make sure you're on familiar territory. This one right here, take with the first one there on the left. The hydrogen, which is the lowest priority group, that's back. That's where it belongs, right, if you're going to call something R or S. So the next thing you'd be doing is looking at setting priorities. And so you've got these three groups right here. And the top priority would have to be this one, right? The first atom out is oxygen, whereas it's carbon, both these spots right here. There's number one. This would have to be number two because that carbon has 2 bonds to oxygen. This one's got only 1 bond to oxygen. So in going from 1 to 2 to 3, you'd be turning your steering wheel to the right as you drive around, and what would you call that? R or S? R. Yeah. Okay. R and S, we're not making a big deal out of that, but I just want to make sure that you are clear that the same old rules apply. This is an organic compound. Same structural terms, discussions, all that, it's still very much intact. The world would be okay. We could use R and S if all carbohydrates had 1 stereocenter in them. But the most interesting ones have like 4, or at least 3. So just R and S is not going to work. You'd have to come up with this long list, R, 1R, 2R, 3S, 4, it would be this long list. So we're not going to go there. Here's the convention. We're going to use something that instead of drawing it with a bond line drawing like this, we're switching to these things right here. And does anybody know, I'll get you a little story about each one of these. These are, I'll say something like, these are regular tetrahedral drawings. And you could have seen something like this in your first organic class. And that's okay too, but the more of these you deal with, the quicker you need to be able to handle them and focus on what's most important. Anybody in your first organic class have this version of things? I know Kaylee did. Okay, do you remember the name for these? Does this look familiar? Fisher projections. Your organic class may or may not have had it. Okay, so that's a lot simpler. The cross right here, which is the abbreviation, that's where there's a chiral carbon. Now these like that one's not chiral, that one's not chiral, you don't mess with them. Okay? They're just stuff out there, and you're interested in emphasizing this. And instead of looking at R or S and trying to figure out what's pointing forward and what's pointing back, you're looking at only one thing. In glyceraldehyde, you're looking at the OH, which in this case matters because it's on the right. In the other one, it matters because it's on the right, or on the left. Those two facts are the only thing that you're looking at to talk about the configuration here. Okay, this one you call D, and this one you call L. You can, it's not far off if you remember L for left. And D technically means dextro, but I don't know if that helps you remember it or not. Okay, both of these right here are what's called not absolute, but relative configuration. And it can be used for much larger monosaccharides. Okay, that's the shorthand that we will use. Now what you want to keep in mind is the relationship that ties the whole side together. So I guess you can even start that up here. Compared to each other, this will be an organic exam class question. Put both of them here. These two compounds are, choose the answer, constitutional isomers, stereoisomers, not isomers of each other. What do you think? Compared to what are these? Are they constitutional isomers? No. They've got the same formula, but they also have the same connectivity. You can't say they're strung together differently. So what kind of isomers are they? They're definitely stereoisomers. And you can home in even more closely than that. Compared to each other, they are mirror images, and that makes them what? They're enantiomers. Okay, now the same is true down here. We haven't changed any relationship just by coming up with a quicker method of drawing things. What you set up here in more detail is also true down here in abbreviation. So these are still enantiomers. They are different molecules. Your body would know the difference between the two of them. And I guess the way to finish up the story right there is to point out one more important fact about just the configuration itself. All naturally occurring carbohydrates are these. There are no naturally occurring ones that I know of, like this. I'll put that in quotes. At the beginning of my career, I used to report the party line that all amino acids had a single natural configuration. By the way, it's the opposite of sugars. The world has handed us D sugars and L amino acids. Go figure. Okay, so I used to say all amino acids have the L relative configuration. So not long after that, here came a finding that proved that all wrong. All of the naturally occurring amino acids that you find incorporated into proteins follow that rule. But wouldn't you know, here's some neurotransmitter. I forget exactly what function it has and in what organism and what tissue and so forth. But it's a natural amino acid that got isomerized from L to D. Well, you watch. The minute we say something like this, there are no natural L carbohydrates, somebody will find one somewhere for some purpose. But learn this big split, I guess. The ones that we deal with in this class are going to fit this rule. They will all be D. All right, now that's a little bit of a basis for thought right there, because that's a signal to you that if there is such a thing, if you could synthetically make such a thing as these guys right here, what might they be like if you use them biologically? I remember when I was an undergrad, there was a company that had come up with an L version of table sugar. The natural table sugar that we eat, I eat too much of, is made of sugars that are like this. Okay, so this smart company says, aha, if we come up with the enantiomer of table sugar, the enzymes in the body won't recognize it. it won't be metabolized. It'll just be excreted as a weight product, and you can get the benefit of sweet taste to your food without gaining weight. That's everybody's dream, right? I don't know that they ever got it as far as commercialization, but they gave it a name. They called it levulose for that L right there in the configuration. All right, there may be other uses out there for exploiting stereochemistry in a smart way, but for us in this class, okay, we'll stick with this part of the world right here. That's the way things naturally come to us. All right, so let's move on to longer ones, and longer than 3, that is. And it's not hard to give them their own Fisher projections, but let's kind of work our way down to that point. What would you say about this compound right here? Well, it has 6 carbons, so it's a hexose. It looks like the functional group in it is an aldehyde, so you would say that this is an aldose. Put the two of them together, and you have the term aldohexose. So there's 2 pieces of structural information in that term alone. It is both an aldehyde and a 6-carbon monosaccharide. This drawing here is, I'm going to stress again, this one is drawn without stereochemistry. But there is information there. So here's functional group. That one is not chiral. And the last one, number 6, is also not chiral. But that does leave you 1, 2, 3, 4. Carbons 2, 3, and 4 as stereocenters. So carbons 2, 3, and 4. This one right here has 4 chiral carbons, and for that reason it has, and here's another organic review. How about if you tell me how many possible stereoisomers? Anybody remember a formula? Okay, it has 2 to the 4th possible stereoisomers. So what would that be? 16? There are 16 molecules that have that formula and that have this method of stringing all parts together, and all 16 of them are different from each other. Only 8 of them occur in nature because we're cutting out half the possibilities in the world. All right, so here is one possible. Let me stress that it is just one. This is just one possible stereoisomer. That would be 1. And the other one is not just any, not just random. You have your hand up, Steve? Yeah. Oh, no, you're just feeling free. Good. All right. So, let's get our numbers in place here. 1, 2, 3, 4, 5, and 6. There they are. And here now we're moving from a stereo view of one isomer to a different stereo view, this one being the Fisher projection. So here they all are again, 2, 3, 4, 5, and 6. Okay, the only thing that happens right here, remember, is that you're redrawing. Same here. So no reaction, nothing more fancy than taking the same compound and redrawing it. Okay, and to be consistent, let's get all these numbers. So you want to start with your, give the lowest possible number to the carbonyl group. In this molecule it's number 1, in others it might be number 2. And go down the list so that you're keeping track of all carbons. Okay, now notice what has happened here. these two compared to each other, these are mirror images, so that's one point. And the more precise way to say this is that they are enantiomers. Okay, so I guess one thing to look for in convincing yourself and kind of keeping track of this stuff is how would I know, just by looking at these two structures here, how would I know that they are enantiomers? Well, you just look at all the chiral carbons and make sure they're exact opposite of each other. So like on this first one right here, carbon number 2, it's out versus back. Go to number 3, back versus out, and so forth. Each one of them is perfectly the opposite of the other. And that means that once you get down here, if you've turned them on their side, the mirror plane should kind of split between the two of them so that close to the mirror is close to the mirror and far from the mirror is the same. I'm going to go down this list. Look where your OH is in the first structure and where it is in the second one, close to the mirror. The next one, oh, that one's farther away from the mirror, so same thing here. Number 4, far from the mirror in both structures, and number 5 puts you back close to the mirror in both structures. So these two, again, they're both enantiomers, still both enantiomers. And let's make sure that we give the name that we know how to give. And again, you're focusing on just the last OH group, the OH group on the last chiral carbon. And this one is D. This one is L. And what you've done by drawing this is you've created just one of the possibilities. This one is, again, just one possible stereoisomer out of 16. You can make that exact same statement about the other one. This is just one more possible stereoisomer of 16. Okay, one of these is found in nature, the other is not. Okay, in fact, that first one is the natural conformation of glucose. Now, just to check you and make sure that you're in the right place in your mental map and not headed toward getting too confused here, here's what I need to probe with you. Take carbon number 1. And I'm not going to draw this to confuse things, but let's just talk about it. Let's say that, and this one right here, the second one, let's say that I had flipped the OH and drawn it over here and the hydrogen here, and everything else was the same. Okay, they would certainly be stereoisomers of each other because the only thing that would change there at carbon number 2 would be where things pointed in 3D. I didn't give anything a new home, just where they pointed. There would be a term that would describe the relationship between those two stereoisomers. They wouldn't be enantiomers, right? Let's say in one of these, I, again, put a hydrogen over here and the OH here. I change one of them but not the other. They're still stereoisomers of each other, but what was the term for that other kind of stereoisomers? Diastereomers, yeah. The world is split into two. The world of stereoisomers is split into two categories. So we've drawn two cousins here that are enantiomers of each other, but we've left out 14. Right? There's 16 of these structures that we could draw, and they would all be that formula right there. They'd all have that structure. These two are enantiomers of each other, but there's a whole world of 7 more out here that are diastereomers compared to this one. Okay, so we're just funneling down on just a little part of the list right here. Out of the list of 16, can you imagine how long it'd take to draw that whole thing? They're all different molecules, and even cutting the world in half so that you have 8 of them with a D configuration, there's still plenty of room for biological diversity just by flipping around the remaining ones. And that will make the difference between like galactose and glucose. It's the direction that one OH group points. We're headed there eventually, but that's all we're talking about to make a big biological difference. Whether things are on one side or the other, oh yeah, matters a lot. Okay, so let's do just a little bit more, and you'll need some time to review for your exam, so try to break a little bit early for that. All right, now we're going to something much closer to the real world. Up to now, the drawings have all been linear, but in the real world, most of them don't exist in linear form. So you can draw them. Again, I'm going to draw just for reference. You can draw them kind of like this, and I'll use a Fisher projection, and let's put in the same number of carbons that we've got on that example we just went through. So like 4 stereocenters and a last one. So here's carbon number 1, 2, 3, 4, 5, and 6, like that. So I'm going to go for one that's pretty representative. So let's say you've got 2 OHs arranged like that. And for some variety, we've got one on that side. And then this last one right here, on number 5, has to be that way in order for it to be a D sugar, a D monosaccharide. All right, the only other allowable partners on these carbons are hydrogens, so you put them in last. What you're looking at here is, to begin with, the linear form, and that fits with the functional groups that we have said have to be in a monosaccharide. It is a polyhydroxy aldehyde. This one's an aldehyde. Could be a ketone. But this thing is in equilibrium with a version of the molecule that looks like this. I'll try to draw this kind of big. You're looking at a molecule that has that as its framework. It's a 6-membered ring. So that much matters. And without cluttering it up here too much, we'll dip back to your organic just briefly. I'd like to think this is good news. You probably spent more time than you wanted to thinking about conformations of 6-membered rings, right? And where things point. Everything you learned is okay for biochem. The only difference is, in place of one of the carbons in the ring, you've got an oxygen. It won't have 2 partners on it, it's got 2 lone pairs. But other than that, stuff you learned about 6-membered rings fits here pretty well. They're nice and stable. That's why they form in the first place. So we're headed into pretty familiar territory. So the 6-membered ring, and within that, one of your ring atoms is oxygen. And when you're all done and drawing it in a 6-membered ring form, all the carbons still have an OH. Now, I say still. Probably shouldn't say still, because one of them didn't even have an OH to begin with. Okay, so let's lay this out in numbered form here. And I'm going to start up on this end right here and add the one carbon that you may have gone, well, I only see 5 carbons there. Yeah, there's one more. 5 of them wound up in the ring. So let's start with it. And that one you're looking at is number 6. Number 6 is kind of stranded out there at the end because it was not in the right position to bite number 1. So let's trace our way back to number 1. So there'd be 5, there's 4, there's 3, 2, and 1. Okay, now here's what you want to be thinking about. Each one of these carbons still has to have its old partners, the ones that didn't participate in the ring cycling reaction, and I'm drawing the H's and the OH's up and down pretty carefully here. And before we go too much into the weeds there, here's what you're thinking about. We've got a linear form, and it is in equilibrium with a cyclic form. Now, this equilibrium arrow right here is maybe not drawn totally accurately, because the real world has like 99% of the cyclic form, maybe 1% of this. So if you buy the stuff as a white powder and dump it out of the bottle, you can, I don't know if I should say you forget about this, but it's all this. So the challenge is to know how to get from one side to the other. And I think what I'll do is just wait until tomorrow or Friday, so we start talking about how you get from this to this. In between, I'll say that you must be precise in tracking your carbons. The things that point right and left in the linear structure have to point either up or down precisely in the ring structure. And that's how we track stereochem. Okay, I think we'll call it done there, and set these exams out.